click go and
away we go. Okay, kia ora tātou. For those of you who don't know me, my name is Louis Schipper. I'm a biogeochemist working here at the University of Waikato, and I see a lot of faces that I suspect are not from the university, so a very big welcome to you. Thank you for coming along. This is a series that is a relatively new series of the Environmental Research Institute, and so we're very lucky today to have Chris Freeman from Bangor, Wales here. Chris and I met, we were just talking back in about 1996 or so when we were fairly fresh out of our PhDs in Perth. I don't think that's quite the right one. We'll find another one. We met in Perth at a conference on wetlands, and ever since then our paths have kind of crossed back and forth. And Chris is probably best known, or at least one of the main things that he's known for, is the enzymic latch theory, and I think we'll hear more about that today, and also observations of increases in dissolved carbon coming out of aquatic ecosystems. Fingers crossed.
Indeed.
And that work has led to several papers in Nature through time, so it's had significant impact. Is it that one there?
Should be that one.
Close this one first.
Yeah, I think we may have to go on offline just for a minute so I can download another copy of this.
Have you got it? Whereabouts have you got it?
It's beyond the cloud storage for Bangor, so I can get it from there if necessary.
All right, we might just pause this for a moment.
I've been recording the whole time.
Oh, no, I did that. Right. We'll sort that out later on. Okay, folks, thank you very much for your patience. We have beaten technology. I'll keep it short. Let's go. Professor Chris Freeman. Thank you.
Well, thank you, Louis, and thank you all for hanging around in that rather disturbing moment there where we wondered whether we were going to see anything at all. But great to see that we've got something at least. So what I'm going to be talking about today is something called the enzymic latch, and one of the ways that we may be able to use this mechanism called the enzymic latch in all of our lives going forward. So here's a picture of a typical Welsh peatland, just a few miles away from my home university in Bangor in North Wales. And I've done a lot of work on these wetlands out there. As this work was going on in the very early years, something quite unusual was found in a peatland quite close to this one. And basically, in that peatland, a severed head was discovered in the peat, buried in the peat. The local police were so concerned that they went around doing house-to-house inquiries, convinced that a murder had taken place locally. And sure enough, they went to one of the houses, and the guy who lived there, well, his wife had gone missing about 5 years earlier, and they put two and two together and charged him with the murder of his wife. And the guy actually confessed to this murder. But while all this was going on, the Home Office scientists, the government scientists, did some routine tests on this head. And basically they wanted to test whether the head was actually put into the bog in the same time frame that his wife was reported missing. So they did carbon-14 dating, and when they did this, the results were rather surprising. It was actually a head of over 1,000 years of age. So this guy had obviously confessed to totally the wrong murder. Well, you're probably thinking, what has all this got to do with peatlands themselves? And what I'm going to show you as we go through my presentation today is that it has absolutely everything about it. And the reason is that peatlands show something really incredible. What they display is a spectacular failure of something that we would take for granted in any other ecosystem, and that's the process of decomposition. When you put a severed head in a peat bog, it basically doesn't break down, and after thousands of years, you could be in real trouble at some point in the future. So what I'm going to do as we go through this is to look at this rather unusual property. So I'll start off my presentation by explaining a bit about what we believe is causing this failure in decomposition and how we discovered this mechanism that we've called the enzymic latch. And I'll move on to explain what the implications of the enzymic latch are and why it's of significance to all of us here in this room today. So let's think a bit about this failure in decomposition that we've seen there. That's clearly an interesting scientific topic to do research into. And basically, we sort of thought the best way of tackling this is to go right back to fundamentals and to look at what it is that brings about the normal process of decomposition. And as we all know, it's all about microorganisms. Microorganisms break things down ultimately into carbon dioxide, but they have to have this rather important ability to be able to achieve that. And that ability is the production of extracellular enzymes. The extracellular enzymes take the high molecular weight material, like the severed heads in the outside world, break them down into small, low molecular weight chunks that they can then get into the microbial cell and allow the complete process of decomposition to take place and discover why it is really that decomposition doesn't take place very effectively. In other words, if we can understand what is controlling the production of these extracellular enzymes, we can understand this whole unusual process that we can see in the wetland environment. Now, normally, in most ecosystems, when we want to measure enzyme activities, it's actually a very simple process. We just use something called a model substrate. It's a chemical compound consisting of two halves. Half of it is some kind of marker, and the other end is a target. So take, for example, an enzyme like phosphatase, which is an enzyme that breaks phosphate off of organic matter. The marker would be some kind of chemical that changes its properties when its bond between that and the target compound, the phosphate, is broken. So an enzyme comes along, breaks that bond, the properties of the marker compound change, and we simply measure the rate at which that marker is released into the chemical solution to get an idea of how quickly the enzymes are operating in these wetland environments. Sounds very simple, very straightforward, and certainly in every other ecosystem, this has been a standard approach that everyone has been able to use very effectively. So whether you're looking at agricultural soils or the depths of the oceans, this is the way that you would do it. Came to try this in a peatland, and it just didn't work. And part of the reason for that is that when we look at the water in a peatland, it tends to be this rich, deep brown colour. Now, can you imagine that if you are trying to measure a change in the properties of a solution, that marker compound abundance, against a dark brown background, it becomes very difficult to measure a colour change. It becomes very difficult to measure fluorescence release. So, in a way, that's part of the reason why it was so difficult to measure enzyme activities in these environments. It's because there's so much in the way of these interfering phenolic compounds in the soil solution. These compounds are phenolics. You may know them by other names like tannins or humics or polyphenols. Essentially all the same sort of thing, these brown coloured chemicals that are so abundant in this type of wetland. Now obviously we realised that if we were going to stand any chance of working out what was controlling the enzyme activities in this system, we needed to find a way that we could get rid of that brown colour and study the release of the model substrate's target compound without that interference. And after a couple of years, we managed to come up with a nice technique using high-performance liquid chromatography, which basically separates out the interferences from the marker compound that we're interested in measuring. So this slide just shows how these are showing the marker compounds coming out of the bottom of the column there, and there is that compound that we actually want to measure, comes out of the column after all the interferences. It meant that for the very first time, we could measure enzyme activities in these peatland environments. And clearly, once you've found a way of doing this, this gives you the opportunity to open up this field to try and understand what it is about wetlands that makes them so special when it comes to the process of decomposition. Well, the first thing we tried in these wetlands to try and understand why they're so different to normal terrestrial systems was to go out to the field and dig up cores of peat that we could then bring into the laboratory and basically simulate drier conditions in those cores of peat. And the basic idea is you make it more like a dry land, and you'll be able to see what it is about the properties of that peat that changes as the conditions become drier. So what do we see? Well, here we can see enzyme activity on the y-axis, a period of time of this experiment taking place. The wet is the control there, showing relatively low enzyme activities. And as we lower the water table and make it gradually drier, you can see that there is more in the way of enzyme activity. So in theory, it all seems to be tied together quite nicely. We've got this system where enzyme activities become more active when we make the wetland more like a dry land. You'll notice that for the first time in my talk today, I've mentioned specific types of enzymes, and what we're looking at in this particular slide is hydrolase enzyme activities. And hydrolases are the important ones to study, basically because hydrolases are the main enzymes involved in decomposition. They are the enzymes that solubilize all of that high molecular weight material and make it available for the microbes in the system. So it all seems to be working well so far. I guess the next link in the chain is showing that when you stimulate the activity of the enzymes, you also increase the amount of the ultimate end product of decomposition from the system, which is the release of carbon dioxide. So here we're looking at CO2 production. Here's that same time period. You can see the lowering of the water table here, the control again, and this line here shows what happens to carbon dioxide release. Very clear evidence that when we make the conditions drier, we not only turn on the hydrolase enzymes, we also turn on the production of carbon dioxide, something that we wouldn't normally expect to see at this sort of level in these peatland systems. All looks very simple and very straightforward, and I remember submitting these results to my first journal in this topic, and quickly getting the reviewers' comments back saying, well, this isn't very interesting. We already know this stuff. You know, we know that you make a wetland drier and it releases more CO2. And the reason is, as these referees pointed out, microbes are more active with oxygen present. And, of course, enzymes, they need oxygen as well. The only problem, though, is previously nobody had actually measured these enzyme activities in the wetlands. So really nobody actually knew that that was what was going on here. It was all just an assumption. And at the time, most of my funding came from the Royal Society in the UK, Britain's Academy of Science, and they have a motto that's been around since the very early days of the Society, back in 1660, when Sir Isaac Newton was one of their founding members. Basically, that motto is nullius in verba. Take nobody's word for it. And that was actually a very apt saying here, because whilst everybody already knew that this was going to be the case, in actual fact, nobody had made those measurements, so we really need to check out those assumptions. So the first starting point for checking out those assumptions was to look for other ecosystems which are depleted in oxygen, just like they are in peatlands. One of the things about peatlands that we all know is that when the soil is waterlogged, the oxygen can move into that soil 10,000 times more slowly than it would do if oxygen was just diffusing in the air. So it's very difficult for oxygen to get into these peatland environments. Because of that, the conditions are anaerobic, and that is clearly a type of environment where we can look for comparisons with other systems. And the two most commonly measured ecosystems where people have looked at anaerobic conditions are anaerobic sewage digesters and the rumen, an anaerobic stomach compartment in sheep and cattle. So, in theory, what this should show is that decomposition is actually very slow in these systems. But when we look at, for example, hydrolase activities to see whether or not hydrolase activity is low in the rumen of a cow or in an anaerobic sludge digester, completely wrong. Despite the fact that there's no oxygen present, hydrolase enzyme activities are very, very high. If we look at decomposition, if we look at microbial metabolism, again, we find that in both of these systems, the activities are very, very high, despite the fact that there's no oxygen present. So this idea that this was a very simple observation that we've made just didn't seem to tie together, because clearly there are places other than peatlands where anaerobic conditions does not shut down the process of decomposition. So the big question is, if it isn't oxygen that's controlling it, what is it? Of course, the next stage in this process was to go back and do a few experiments to look at the enzymes themselves and to see what the enzymes did in the presence and absence of oxygen. So here we've got the anaerobic systems, and here the aerobic, the oxygen abundant systems. And what you can see here is that while for these particular enzymes there may be perhaps a little bit of an increase with sulfatase, perhaps a bit of a decrease with beta-glucosidase, maybe a bit of an increase with phosphatase, these are nothing like the differences that we would expect if oxygen was an on-off switch in this system and regulating what was going on. But first we thought, well, maybe we've just picked the wrong three enzymes. These are, if any of you measure hydrolase enzymes, you'll know that these are the three cheapest that you can buy. Perhaps we just made the big mistake of just going for the cheap ones. Tried a whole range of different hydrolase enzymes, and all of them show the same thing. It doesn't really seem to matter whether oxygen's present or oxygen's absent. Hydrolases seem to work quite effectively. But then one day, we decided to look at another enzyme, an enzyme called phenol oxidase. And that particular enzyme responded massively to the increased abundance of oxygen. So there's clearly something going on here, something that can give us clues as to what's going on. In many ways, it seems very simple and straightforward, and maybe you'd conclude that the phenol oxidase is what's responsible for the decomposition. In actual fact, it can't be that simple. There's another question in all of this. If the hydrolases don't mind whether oxygen's present or absent, why then is it that when we made the wetland conditions drier that we saw the hydrolase enzyme activities go up? There's clearly a bit of a paradox in this, and it takes quite a while to try and unravel what's going on. But believe it or not, already, in what we've told you so far, all the clues are already there. So let's have a look at the very first clue that helps us to explain this. The first clue comes from looking at essentially what phenol oxidase actually does. phenol oxidase is actually one of the few enzymes able to attack that phenolic material, the brown colour that's so abundant in these peatland environments. And what we're showing on this graphic here is that as we lower phenol oxidase activity, you can see that wherever phenol oxidase activity is low, we have wherever phenol oxidase activity is high, we have low levels of phenolic compounds in the environment. If for some reason the phenol oxidase activity is low, then phenolics accumulate. So phenol oxidase is an enzyme which can clearly break down phenolics in the environment. The only problem, of course, is that the phenolics are quite a small proportion of the total amount of organic matter that's present in a peat bog. Most of it is non-phenolic compounds, so it does seem a little bit surprising in some ways. So what other clues have we got? Well, let's have a look at what happens to phenolic compound concentrations when we make the conditions drier and stimulate the phenol oxidase in the environment. Basically, what we can see here is over that same 15-week period of drier conditions, as we lower the water table and make conditions dry, one of the most dramatic effects we can actually see is the loss of phenolic compounds from the soil solution. The water becomes much clearer when we dry the wetland out. So another clue that gives us an idea of what's going on here, but certainly doesn't give us an instant explanation of what's going on. The final clue, and the one that gives us the biggest pointer as to what's going on, comes from looking at what the effect is of those brown phenolic compounds on hydrolase enzymes. Remember, the hydrolases are the main enzymes involved in decomposition. So, in each of these slides, what we're looking at is phenolic compound concentration on the x-axis and hydrolase activity on the y-axis. And what you can see there is a classic example of inhibition. Basically, the higher the phenolic compound concentration in the wetland, the lower the hydrolase enzyme activity because of the inhibitory properties of these phenolic compounds. So believe it or not, you've got all the clues that you need to understand what's going on, but I always think at this point it's an idea to step back and to try and understand it from its most fundamental level. So the most fundamental level, what does decomposition involve? Well, it involves microbes producing hydrolase enzymes, extracellular enzymes that they release out into the environment, and those hydrolase enzymes will attack the organic matter out there in the environment, with severed heads or whatever else it is that you've got out there in the environment, and it will release from that material available nutrients, so organic substrates, low molecular weight organic substrates, and inorganic materials as well, essentially creating large amounts of resources for the microbes. And those microbes can then produce even more hydrolases, bringing about this runaway process of decomposition that we're used to seeing in every other ecosystem. Now, we've said this doesn't occur in these wetland environments, so what's going wrong? We used to think that it was simply down to oxygen, but as we now know from what we've looked at so far, we can take the oxygen away completely from this story and it makes no difference. If we look at the rumen of a cow or an anaerobic sludge digester, this same process, this runaway process of decomposition, seems to work away quite happily without any constraints whatsoever. So the big question is, well, what is so different about these peatland environments? And that difference is that abundant brown liquid chemical that we've looked at, the phenolics. The phenolics, as we've seen, are inhibitors. They will inhibit the ability of the microbes to synthesize new hydrolase enzymes, and if any hydrolase enzymes get out there into the environment, the activity of those hydrolases is prevented from releasing available resources to feed the microbes. So effectively, those phenolics shut down the entire process of decomposition. In many ways, this is the enzymic latch on decomposition. This is what stops that runaway breakdown and decomposition of any severed heads that you want to dispose of in any English peatlands. Now the thing is, we know that if we make the conditions drier, we can reverse this. So what do we need to do that? Well, in a peat bog, the one thing that we are going to need is some kind of chemical that will remove the phenolic inhibitors, and that chemical is phenol oxidase, the enzyme we've just been looking at. And phenol oxidase is one of the few ways that we can actually get rid of phenolics in an intact peat bog system. Unfortunately, there is lots of this phenol oxidase actually in peat bogs. The only problem is it doesn't do anything. And the reason it doesn't do anything is because there's no oxygen, because it's so difficult for oxygen to diffuse into the waterlogged peat soils. So to really bring this together, it's only when oxygen comes into the system through drainage or a drought event that this triggers the phenol oxidase into action, and the phenol oxidase destroys the phenolic inhibitors, and then we start to see this runaway decomposition process that we would take for granted anywhere else. Well, we were lucky enough back in 2001 to get this published in Nature, and it's been the cornerstone of much of the research that's gone on in my lab ever since. So why is this important? Well, one thing about peat bogs is they have plants growing in them. And as we all know, plants take carbon dioxide out of the atmosphere, they convert it into solid carbon-rich biomass, and normally at the end of the life of that plant, it would die and decompose and release that CO2 back to the atmosphere. But thanks to the enzymic latch, that's not the case. Decomposition is very slow, much slower than the rate at which the plants grow. So as a consequence, there is now a huge build-up of carbon stored in the world's peatlands, an area that covers no more than 3% of the planet, but it stores almost as much as the CO2 content of the entire atmosphere. So that's a huge store of peat. And part of the reason that we got this published in Nature was the realization that if for some reason this enzymic latch was to fail, then you could potentially release all that CO2 back into the atmosphere. And already there is some evidence that the enzymic latch mechanism is weakening. It's weakening because of climate change, creating more drought events, for example, but also because of some of man's more direct impacts on these wetland ecosystems. This particular study, another paper that we got published in Nature, which basically looked at the age of carbon that was coming out of peat bogs in Malaysia. And we were comparing oil palm plantations in Malaysia that were replacing the pristine vegetation of the peat swamps that were out there. And the interesting thing that we found was that the carbon that was leaking out of that wetland was around about 4,000 years old. A guy called Tim Jones in my lab discovered this in one of our samples, and at the time that was the highest age for a naturally preserved, non-frozen source of organic carbon that had been preserved for such a long period of time in a wetland. And here we are carrying out drainage activities in these swamps to produce oil palm plantations, essentially for biofuels, and actually releasing large amounts of ancient stores of carbon back into the atmosphere. All sounds a bit depressing and negative, so I like to switch my talk over a bit at this point and say that it's not all bad news. There must be some positive ways that we can use this new knowledge of the enzymic latch in these systems. And one of the ideas I had quite early on was well, maybe we could use the enzymic latch as a way that we can preserve food materials. I thought, well, this could be a good patent for the future, make a bit of cash for the lab. In actual fact, I was jumping the gun a bit because it turns out that I have it on assurance from many people that the Vikings were already doing this 1,000 years ago. They were collecting fish, they were burying them in the peatland and preserving them to consume as a resource later on. So maybe as a new food technology, they're preserving foods that doesn't actually need refrigeration. Is something that may be a bit further off in the future, especially when you try to get people to sample some of this material. This is another example of preservation that has taken place over the years. And over in Ireland, they used to bury butter in the bog as a way of preserving it. And here's some bog butter, as it's known, that's been preserved for several hundred years below the ground. Interestingly, we've always found it very difficult to persuade people that they ought to give it a try, but maybe if things become difficult in the future, that might be something we'll be looking at. The big thing about this is it doesn't require refrigeration, so no energy involved in the preservation at all. Food technology clearly out. Perhaps something that's more promising though is something we've been looking at is the way that we could potentially use the enzymic latch in wetlands used for water pollution treatment. And over in Wales, there's a bit of a problem in some of our drinking water reservoirs in that they get lots of nitrogen and phosphorus into them. And this is an idea we had of creating floating enzymic latch treatment wetlands where you carried these structures out into the middle of your reservoir. The plants take up the nitrogen and the phosphorus. If the enzymic latch is intact, they will essentially retain that nitrogen and phosphorus for long periods of time, essentially acting like a biological filter, treating the water using natural techniques. But the real benefits of this is that at the end of that period of capturing the nitrogen and phosphorus, you can actually tow the wetlands out, you can carry it to some farmers' agricultural land, and as the water drains away, that allows oxygen in, the enzymic latch is opened, and you can release that nitrogen and phosphorus fertiliser in somewhere where it can actually be used as a source of valuable fertiliser. So maybe that's one area where there is something a bit more positive than spending too much time focusing our attention on the depressing prospect of climate warming Armageddon as a result of a destabilised enzymic latch. But there's another way that this may be important as well. And that's in this new emerging field of geoengineering. Geoengineering's suddenly beginning to take attract a lot of interest, certainly in the UK, where a lot of people are starting to realise that we seem to be struggling to persuade anybody really to cut down on their carbon emissions, and perhaps we're going to need a plan B, some alternative way of reducing global warming on the surface of the planet. And so places like NASA have been considering space-based mirrors to reflect away some of the solar radiation. We've got a variety of afforestation schemes suggested for the future, and all of these are attracting considerable interest. It's all about looking for a quick fix, essentially, that will allow us to continue our normal economic activities without essentially having to reduce our fossil fuel consumption. Probably not something we really want to be considering, but it's there as a plan B in the background. As I said, solar radiation attracted lots of interest. We've mentioned the space-based mirrors. Those not in Oxford and Cambridge University. There's a group that have been looking at using tethered balloons and a long pipe to pump up and spray aerosols of sulfuric acid into the stratosphere. Or you could put it out in plumes behind jets as you're flying along in the atmosphere. Sounds good, but it's obviously going to be quite expensive. And the other thing is it doesn't really address the source of the problem, all that carbon dioxide that's there in the first place. It also doesn't solve problems like ocean acidification that we all ought to be thinking a lot more about now. So my preferred option is to look at the other side of the story, which is all about carbon capture and storage. And there are physical ways of doing this, essentially creating artificial trees to soak up the CO2 out of the atmosphere and keep it locked up, or the ones that I suspect most of us here today are more keen to focus on, and that's the biological techniques. Some have suggested ocean fertilisation with experiments where iron has been added to the oceans to stimulate the algal growth, to get them to take carbon dioxide out of the water. Others have looked at afforestation, some have looked at biochar, but each of these has their concerns that are raised with them. Ecological concerns, concerns with afforestation about longevity. Trees only have a certain amount of lifetime. If they die, what's going to happen then? They will decompose, release the carbon dioxide back into the atmosphere. Others have considered biochar. That's more like it. It stabilises things on millennial timescales, but it also requires a lot of energy to input to bring about the storage properties. So basically, what we've been suggesting is that we could learn something from the peatlands, because here is a system which has accumulated a huge store of carbon in a very small proportion of the planet. We know that wetland plants will soak up 0.5 kilogram potentially of carbon per metre squared per year. Sadly, we know that they don't do that very effectively at the moment because there's still a lot of decomposition in the surface aerobic layers. And the idea is that if we can find a way of getting more of that carbon directly into the long-term storage, we've got the potential to store carbon on millennial timescales that will be very valuable for the planet in the future. So basically what we're interested in doing is showing that there are ways that we can use the enzymic latch to achieve this. We've always thought that there was a way of doing this by making wetlands waterlogged and wet. There's no problems about that, but we now know there are also other ways that we can do that. As we look in more detail at the enzymic latch mechanism, we've discovered that at each of these different points within the enzymic latch mechanism that we've looked at so far, there are different aspects that we can modify in some way by changing the environmental conditions that the microbes and these enzymes are exposed to. So just to take a couple of examples, available water is one way that we've mentioned quite a lot. This slide shows moving along a gradient from very wet soil to very dry soil. And you can see that for most wetlands, if you make conditions drier, it actually makes the enzymic latch weaker. It allows decomposition to take place, and phenol oxidase activity is high. If we continue to make it drier still, we can actually lower the phenol oxidase activity further. Oxygen itself, or water availability, begins to constrain the decomposition process and the effectiveness of the enzymic latch. So if we want to store carbon, we've actually got two choices. We can make it wetter, or we can make it much drier. Both will have the same effect of making conditions drier. And here we can see in some results that we published last year in Nature Communications, the effects of pH on the enzymic latch mechanism and the phenol oxidase within this system. And what you can see is that as pH rises, you get more active phenol oxidase. If you want to store more carbon, you lower the pH. So across these different approaches that we've been looking at, there are two different approaches. We can look at ways that would strengthen the latch in the wetland itself, and the latest approach we've been taking is to look at ways that we can increase the range of land types that are influenced by the enzymic latch. So transplanting the enzymic latch from the peatland to other ecosystems. And we'll start off with that idea of transplanting elsewhere. This is something that's been looked at by the University of Texas, for example. They've been looking at low tillage agricultural. Low tillage agriculture is all about not introducing oxygen into the soil by ploughing. So exactly what we want to do if we want to keep the enzymic latch closed. They've also been looking at adding phenolic-rich residues with this, and the two combined together seem to be very effective at storing more carbon. So it does seem to be possible to transplant this enzymic latch mechanism outside the wetland itself to a degree at least. Within the peatland, we thought, well, perhaps we could grow these forests that we've all been talking about in the forestation schemes, but then relocate that timber to the wetland and bury it in the peat. If we do that, we know from the severed heads that we've stumbled across that will store that carbon in a stable form for thousands of years into the future. Seems like a good idea, quite expensive to do. Not sure that's going to catch on. Might be a bit disruptive for some of our remaining undisturbed peatlands as well. We've also been looking at ways that we can increase plant productivity and shift the communities within the wetlands, essentially looking for ways that we can have plants that are growing that don't introduce oxygen into the soil, for example, or increase the rate at which the plants will grow. Seems to work on one scale. You can get the plants to grow faster, but you've also got this complication that quite often when you add nitrogen and phosphorus, it stimulates decomposition as well. So got a way to go with that one. One of my favourite ones that we've been looking at in Bangor, I work in a department dominated by zoologists, and one of the fiendish ways I've persuaded students to take on biogeochemistry is to tell them all about this animal here, the beaver. And we've got this fiendish plan in Bangor to rear up an army of beavers. 400 million beavers is our target. Releasing them out there into the world so that they can build dams, create more peatlands across the landscape in a much more cost-effective way than the way we've been looking at, which is installing dams in the environment. And the beavers do all the work for free, and clearly a good thing for the environment. You won't be surprised to hear we've been struggling to get permission to release any beavers yet, but the idea's there, and we're ready. So what about the pH? Remember I mentioned about the pH being a useful way? Well, we've been looking at ways we can acidify the peat with watering cans of sulfuric acid. Clearly, it did actually work. It did slow down decomposition. We realised we needed something larger scale, so helicopters would clearly be useful, but even that could be quite expensive. We realised, well, our colleagues in Oxford and Cambridge, we've got this big balloon injecting sulfuric acid into the stratosphere. That could be quite useful for us as well, because what goes up must come down. We could bring back acid rain. That must be a good thing for the environment. If you're not worried enough about my plans for peatlands in the Northern Hemisphere, perhaps it's not quite so worrying when you're down here, but this really gets people worried up north. So I had this idea that, well, perhaps what we should really be doing is using military techniques to inject across the surface of all our pristine peatlands with sources of phenolic waste, which would not only get rid of that phenolic waste, but it would be injecting phenolic inhibitors across the landscape to slow down the process of decomposition. Not had much luck with getting permission for this on a large scale either. Although we are trialling it in Canada in peatland restoration sites and seeing some quite promising results that this is a very effective way of adding these phenolic residues and accelerating the process of peatland restoration after peat mining activities. And if that's not worrying enough, what always makes people in Europe at least go very pale is when I say, well, my ultimate aim is to genetically engineer sphagnum plants. Restiads, I guess, would be your equivalent here. And the basic idea is we modify the genes in here, which changes the way that they produce phenolates. Essentially, the idea is thanks to my colleague Anil up there, we've actually been very successful in finding a way to turn off the controls on the production of phenolics. So genetically modifying sphagnum plants so they continuously produce an overabundance of phenolics into the environment. And that clearly has some potential if we were to release this genetically modified plant out there into the environment. I've often tried to persuade people that one day we may actually consider this to be quite an acceptable idea. And believe it or not, ever since I started making presentations on this topic, I've referred to a study from here in New Zealand where some social sciences studies took place to look at the acceptability of genetic modification in the environment. And what they found in that study is that there are some areas where there is less resistance to genetic modification than others. That's great to be able to talk about it for the first time in New Zealand itself. Basically, genetic modification for pollution control, nobody seems to be worried about that. They seem to think that's a good idea. Like human gene therapy, again, anything medically related can be good. The one thing that none of us seem to like the idea of, both here in New Zealand and up in the north, is food. Anything that we've got to eat that's genetically modified is not considered a good thing. So my ray of hope for one day being able to release this genetically modified sphagnum into the environment comes from the idea that, well, sphagnum's a plant that nobody wants to eat. Clearly, there's the potential here for us to gain acceptance to release it out there into the environment. Well, I'd like to end my presentation by just pointing out that I accept that there are risks and rewards with everything that I've talked about today. And clearly, biodiversity, there's a major risk of any of this to biodiversity. So clearly we do need to carry out more research to look at the impacts of these changes that we're proposing on biodiversity. Other people focus on the risks, and many people have pointed out to me that the biggest risk of all this is that if that group up in Bangor becomes so successful at locking up carbon, we could actually trigger the next ice age. And that could be quite serious. And I've been trying to reassure people that this will never be an issue, because one thing that we know is that we should be able to remove any chance of that runaway cooling by some of the techniques we've been using in many places around here. Digging drainage ditches, so circular drainage around your peat bog. We're all experts on this. I'm sure that we'll have no trouble at all in finding ways of ensuring that that carbon sequestration doesn't continue to dangerous ways. In other places, fires have been a very fast way that we've found of getting that carbon back into the atmosphere. And an idea from Ireland that we may need to think about is that, in actual fact, what we're creating with this acceleration of the production of peat is a biofuel. And potentially that could actually be very useful for the future as a source of energy. So I'd just like to end really my presentation then by pointing out that whilst the enzymic latch might seem a bit of a depressing subject, there are actually some ways where we can offer a ray of hope. The possibility one day, if we can get people to give it a try, of preserving peat with zero energy costs, no refrigeration, by adding these phenolic compounds to our food. Novel water treatment techniques where we can recycle nitrogen and phosphorus, get it back out there onto the land where we want it. And there's also this alternative approach to geoengineering, locking up vast quantities of carbon dioxide in the peatlands and other ecosystems using this peatland enzymic latch as well. And finally, that final suggestion that maybe what we're doing here is we're creating this vast resource of renewable energy for the future, which could be very useful in the future. So thank you very much for your attention. Thanks.
you very much, Chris. I'm aware that people might have appointments, so if you do need to duck out, please go ahead. But we'll take some questions. I was fascinated to see you take it all the way from the individual enzyme studies through to the ecosystem, and I think this is the first time I've ever heard of somebody sort of suggesting that we could drain peatlands in case there's runaway cooling. That took me by a bit surprised, really. So, do we have any questions for Chris, please? Yes, please.
At some point you mentioned adding nitrogen compounds, but I don't think you discussed that further.
Well, there is some evidence that in lignin-rich peats, that when nitrogen is present, it acts to amplify the inhibitory effect in the system. Some research going right back to the 1980s that suggests that. So the caveat is that it has to be lignin-rich, but it is true that adding nitrogen as well as phenolic compounds that are rich in lignin would actually be an effective way of sequestering more carbon.
With many systems, there's sort of a threshold where above that it becomes, not a good thing. So have you kind of looked into that with phenolics? If you're looking at increasing the phenolics in the ecosystem, have you looked at where that threshold is?
That is a very good point. What we've been doing so far in our studies in Canada is looking at increasing levels of phenolic compounds, and obviously what you want to do is you want to increase the inhibitory effect in the soil, but the important thing is not to become so inhibitory that you completely shut off nutrient cycling, which is important to ensure that the plants have access to valuable nutrients like nitrogen and phosphorus. So that is a very important question. So far, none of the levels that we've tested has actually had a negative impact on plant growth. So in theory, that suggests that there is some way to go before we reach that threshold, and we haven't found a way to exceed it yet. But I wouldn't rule out that one day in the future we may find like an optimum at which you should not add any more phenolics to the system. Thanks.
Any more questions for Chris? Troy.
So it's a very clever strategy, and I think it makes you wonder whether plants might themselves have adopted it at one stage in the past, and it might actually manifest itself as something that we often have declared to be something else, perhaps allelopathy, and whether you could identify any examples of that.
I think that's a very good point, and there is clearly some evidence for that, and the fact that at certain points in the Earth's history we've had dramatic increases in carbon storage, thinking of the accumulation of our coal reserves, for example. It's all accompanied with a dramatic fall in oxygen availability. Could it be linked in with the evolution of phenolic compounds themselves and lignin-containing compounds? I think there is something.
That reminds me a bit of Randy Dahlgren's work, right, on the ecological staircase, where nitrogen's tied up with phenolics.
Right.
And so they're slowing the whole nutrient cycling down, I think. That would be my kind of gut feel, anyway.
I was just wondering kind of where to
Thanks, Troy.
from now, like that's pretty powerful links with, say, olives, for instance, having those really high phenolic compounds, and there's really massive olive industries actually spread out globally. So kind of how do you see your research now actually affecting us on a more day-to-day basis and government changing blah blah?
I mean, I'd like to think that there would be more government support as concerns about global warming become more intense. And there is evidence that does seem to be happening in this last few years. It's very encouraging, I think, that people have been much more keen to switch from fossil-fueled vehicles, for example, to electric vehicles. If if we continue to see the extremes of weather that we've been having in recent years, then I wouldn't be at all surprised if in future we don't see even more interest from, at a government scale, in looking at these alternative ways at which we could encourage more carbon to be sequestered in natural ecosystems. And of course, agricultural ecosystems must be even better, because if we can continue to produce food on the same land that we're sequestering carbon using enzyme latch techniques, then clearly that's a win-win for everyone concerned. So I think if we can go back to this idea of generating carbon credits, which does seem to be coming back onto the political agenda after an initial failure about 10 years ago. I think when there are financial incentives in place to encourage people to sequester more carbon, then I suspect you'll even get the farming community themselves looking for ways that they can sell carbon credits on their land, essentially through enzyme latch techniques. So, yeah, it's there, it's in the future, but that future gets closer all the time.
All right. I'd like to thank Chris one more time, if you wouldn't mind joining me. Have we got another seminar next week?
In 2 weeks' time.
2 weeks.
Yeah, so in a couple of weeks' time, we've got another seminar coming up where Kate McInnes-Ing's coming down from University of Auckland to talk about climate impacts on New Zealand forests. So that'll be another interesting one that probably follows on nicely from Chris's talk. Thanks very much.